All right, let's dive into one of the coolest, most exciting frontiers in all of biology, functional genomics. We're going to talk about how scientists are finally learning to read the book of life and what that means for, well, for everything. So picture this, the turn of the millennium. Scientists complete the Human Genome Project. It was this massive international effort, and what they gave us was incredible, a list of all our genes, roughly 35,000 of them. It was like getting the complete parts list for a human being, a huge, monumental achievement, but it left us with one giant question.
Yeah, what do they all do? I mean, it's one thing to have a list of 35,000 parts for a super complex machine. It's a whole other thing to know how that machine actually works. We had the Book of Life, but we couldn't really read the language yet.
And that was the central challenge, right? We had this massive, untranslated instruction manual, and it was pretty clear that the old ways of doing things, they just weren't going to cut it. Not even close.
You see, the classical approach to genetics was, well, it was meticulous, but it was agonizingly slow. We're talking about scientists studying one single gene at a time, sometimes for years, just to figure out what it did. Now, you do the math. With 35,000 genes, that's not just slow going, it's basically impossible. We needed a totally new approach, something that could read whole chapters, not just one letter at a time.
And that new approach, of course, has a name. So let's meet the detective that showed up on the scene to start cracking this massive genetic mystery. And here it is, functional genomics. The big idea here is to stop looking at one gene in isolation. Instead, it's about understanding the entire system all at once. And it does this by teaming up massive, large-scale experiments with some seriously powerful computer analysis. It's a true partnership between the lab bench and the computer chip. And this partnership is really the engine that drives the whole field. So on one side, you've got the experimental scientists in the lab, just generating these mountains of data about which genes are switched on and when and where. But on the other side, you've got the computational biologists using something called bioinformatics to sift through all that data, find the hidden patterns, and actually predict what a gene does. So how do they actually generate all that data? Well, let's pull back the curtain and look at one of the most important tools these genetic detectives have in their toolkit.
Meet the DNA microarray. You might also hear it called a gene chip. This little device was an absolute game changer. It basically gave scientists the power to see the activity of thousands of genes all at the same time.
The concept behind it is actually pretty cool and surprisingly simple. Just imagine a tiny microscopic grid on a glass slide. Each one of those little squares on the grid has the DNA for one specific gene attached to it. So you can think of it like a giant switchboard for the entire genome, designed to light up and show us exactly which genes are switched on at any given moment.
So how does the switchboard work? It's a multi-step process. First, you take the cells you want to compare. Let's say cells from a cancerous tumor and cells from healthy tissue. You pull out all the active gene messages from both samples. Then you wash those messages over the chip. And wherever a message finds its matching gene on the grid, it sticks. A laser scanner then reads the chip. And what you get is this beautiful, direct, visual comparison, a pattern of glowing dots that instantly shows you the genetic differences between being sick and being healthy.
And the applications for this are just incredible. This kind of tech helps us discover brand new genes. It lets us diagnose diseases like cancer with way more precision, and it's even pushing forward personalized medicine. There's this field called pharmacogenomics, which is all about tailoring drugs to a person's unique genetic profile to make treatments that are safer and way more effective. It's a seriously powerful tool.
But here's the thing. A microarray just gives you a pattern of dots. It's a massive amount of raw data. And honestly, that data is pretty much useless without its essential partner, the computational brain that can actually make sense of it all.
And this is where bioinformatics comes in. Without it, the data from a gene chip is just, well, it's just noise. Bioinformatics gives us the tools to turn all that noise into real, actionable knowledge.
So think of it this way. Bioinformatics is the master librarian and detective. It organizes all these clues into these huge searchable databases. It has powerful search tools that can find genes with similar sequences, because similar sequence often means similar function. It can compare our entire genome to, say, a mouse's to figure what makes us different. And maybe most importantly, it can predict how all these thousands of genes and proteins are actually talking to each other, revealing the complex networks that keep us alive.
Which, brings us to the whole point of all this, the ultimate goal. This isn't just about curiosity. It's about taking all these genetic clues and turning them into actual cures. And this quote from our source material really just says it perfectly. We now have thousands of potential new drug targets we never knew about before. The job of functional genomics is to help us figure out which of those are the best targets, so we can design smarter, more effective medicines instead of just relying on random trial and error. So, at the end of the day, what functional genomics is doing is building a complete, multidimensional profile for every single one of our genes. It's creating this incredibly detailed map of our own biology, and it's highlighting the exact points where we can intervene to fight disease. This is the real foundation of precision medicine. The Human Genome Project gave us the book of life. Functional genomics is finally teaching us how to read it. And as we get more and more fluent in this amazing biological language, it leaves us with a really inspiring question. With this kind of power, what disease will we understand and conquer next?